One of mankind's biggest killers is also one of the tiniest: viruses. Towards the end of the First World War, an infection started spreading around the world. It infected mostly young people and killed many of them. Over the course of 2 years, it infected 500 million people and caused more deaths than the First World War, which was just ending. The virus responsible was an influenza virus, which we know as Spanish flu. Viruses cause a huge range of diseases, and we're exposed to them on a near daily basis. Common infections that we get that are caused by viruses include things like measles, chickenpox, and the common cold. Viruses also cause more deadly diseases like HIV and Ebola, whereas many bacterial infections can be treated with drugs like penicillin. There are relatively few effective antivirals. We study two different things in our laboratory. The first is the structure of viruses, so what they look like at the atomic and molecular level. We also look at how the immune system fights infections and keeps us healthy.
We use a special technique called Xray crystallography. So the X-ray beam fires X-rays at the crystal, and they diffract through the crystal and make a pattern. And all these patterns are collected by a computer, and it's put through a special computer program. And what comes out at the other end is this. So what we have here is the structure of our antibody and the structure of the surface of our virus. And we're looking at very, very, very high resolution. So we can actually see individual atoms on the virus and individual atoms on the antibody and how they're interacting. And this helps us understand how the shape of antibodies mimics the shapes of viruses and understand better how our body fights viral infection.
Another way we have of measuring viruses and how they replicate and infect is by examining how they infect human cells in the lab. So when we grow human cells to see whether or not they've been infected with virus, by looking to see whether those cells have gone green with this fluorescent protein we've taken from jellyfish. So if you look at the image here, what you can see is lots of green splodges, and each of these splodges represents a single human cell that's been infected with the virus. And here you can see that the virus has been very effective, and it's infected all of these cells. Using these jellyfish-containing viruses, we can now study how viruses infect cells, but we can also investigate what things cells do to defend themselves from viruses. So here we have viruses, which are shown here in blue, in circulation, being targeted by these yellow molecules, which represent antibodies. And these viruses are seeking to infect the cell, and they do that by sticking to the surface of the cell, and then the cell takes them up, thinking that this is a useful metabolite. The virus is adapted to be able to leave these small compartments called endosomes to escape into the main compartment of the cell called the cytosol. At this point, the cell can tell and detect it's been infected because of these antibody molecules stuck to the virus. These antibody molecules are recognized by TRIM21, shown here in cyan. TRIM21 recognizes the antibody and delivers a signal to other machines in the cell called the proteasome, which are shown here in orange. And the proteasome are the cell's garbage disposal system. And they recognize the virus as junk and literally tear the virus apart, breaking it down into its component pieces and preventing it from infecting the cell. So the discovery of processes like TRIM21 and antibodies and how they prevent cells from being infected by viruses gives us new information about designing antiviral drugs. We can learn from these natural responses that take place inside all of us in order to make new kinds of drugs that might prevent infection. And they might work in a number of different ways. They might work alongside these mechanisms in order to augment them and make them work better. Or we can borrow those concepts in order to design other antiviral molecules which maybe work in conjunction with this system.
These are just two ways that our lab has been trying to understand virus infections. There have been a lot of discoveries in the last 100 years, but we're just at the beginning.